Welcome, everyone. In the meanwhile, we give also a little bit more time for people to join. And today we are delighted to host Periklis Pantazis, a group leader at the Department of Bioengineering at Imperial College London, where he leads an interdisciplinary research program between developmental and stem cell biology, engineering, chemistry, and optics. Lucky studied biochemistry at the Leibniz University of Hanover, obtaining a PhD in biology and bioengineering at the Max Planck Institute in Dresden, for then moving in the USA as a postdoctoral scientist. Luckily worked at the Californian Institute of Technology and then came back to Europe as an assistant professor at the ETH in Zurich. And since 2018, he joined Imperial, where he's now a reader in advanced optical precision imaging, where his group is devoted to the development and applications of cutting-edge imaging technologies and reagents. So thank you, Lucky, for your availability today, and we look forward to your presentation.
Okay, thank you very much. Let me share the screen. And thank you first for your, Alessandro, for the introduction. Yeah, it's a pleasure presenting today about prime conversion. I do hope that you can see my slides and also my mouse. So prime conversion, the new kid on the block for photoconversion. Some of you might already be aware of photoconversion. So again, yes, I'm called Lucky, so it's the nickname. And this is the Laboratory of Advanced Optical Precision Imaging at the Department of Bioengineering at Imperial College London. Before I come to prime conversion, just briefly, what kind of, let's say, imaging technology do we conceive and also hope to apply now in the near future? One of them I'm going to talk today is prime conversion, which is precise and non-toxic labeling for inferring, let's say, cell state transitions in disease, let's say cancer, as well as also in development. And I'm going to go in much more detail about the technology. 2 points I will not cover today. One of them is GENEPI. This is a genetically encoded fluorescent reporter for mechanical stimuli. This is based on a piezo function. So visualizing, let's say, stretch, shear stress, also in a physiological context. So this is not covered today. And the other one, which is going to come out also soon, is biharmonophores. These are nanoprobes, which give a very strong nonlinear signal. And these ones are not only biocompatible, but biodegradable. So they open up the avenue for clinical applications. And so we want to use them. In this case, they identify cancer cells, but not only for diagnosis, but also for therapy. So take advantage of their light. And this way also, for example, killing the cancer cells in close by. Now, these 2 points we will not cover today. I'm going to talk about prime conversion, and I give you, let's say, I hope so, a comprehensive overview of what has been done so far, and let's say also the backgrounds and applications. So before I come to this, I would like to give you, which has been a decade before, how it led then actually to prime conversion. So, and the idea was really to understand principles of developmental biology. And one of them is, in fundamental question is, how does a single cell develop into complex multicellular organism? And we focus in there on early mammalian embryos. So you see then the stages in the first 3.5 days, starting from fertilization, and then you have then 2-cell stage. So this takes roughly a day, and then it gets a little bit faster and faster to 4-cell stage, 8-cell stage. As you can see in these ranges here, the cells are morphologically indistinguishable. And later on in the process from 8 to the 16-cell stage, where you have the morula, you have a process which is called compaction, where the cells then allocate that's outside and inside with respect to the membrane. This is the zona pellucida. And now these inside cells are presumed then later on to form the inner cells, and the outside cells form now the tefectoderm here. So it's the TE. So the inner cells gives rise to the embryo, and the tefectoderm gives rise into the extraembryonic lineages, so placenta. Now the question, it's still debated, but at that point it was much more debated. Is really how is model of asymmetry being generated. And traditionally people thought this is stochastic, meaning that the first cells at the very early stages can all contribute to inside cells as well as to the inner cells and tefectoderm. And only the physical allocation to outside inside cells lead then really then to the different lineages. And the other one is the biosynchronicity. So this wording actually started to develop in the last 10 years. is that there's potentially some qualities earlier before compaction, where the cells are indistinguishable, that give them some qualities to allocate either to outside or inside cells, be it, let's say, epigenetic modifiers, cell divisions, maybe even mRNA localizations. All this analysis was motivated much more because of C. elegans and other work where they have done actually this analysis. What we introduced actually then as a seminal work is the first time to do quantitative dynamic imaging, stochastically significant, so all the embryos, and this was based on dynamic imaging for transcription factors. So we looked at OCT4, and OCT4 is known to be expressed everywhere, but the mutation later on leads only to effects in the inner somas. So we wondered, hey, is a dynamic maybe an identifier of any, let's say, a propensity to allocate to the inner cell mass of the two-factor derm? So we looked at the nuclear dynamic of OCT4, and we didn't really look at precise interactions, let's say, how long does it stay on the DNA? So we made it in a rough way. So we talked about export, import, degradation, immobile fraction. And so we developed a description of nuclear OCT4 dynamics. So the nuclear population over time depends on import, export, and degradation. And we wanted, of course, to do it dynamically, so imaging in a developing mouse embryo. Well, it's still a molecule of choice is green fluorescent protein, right? It renders all the populations visible. And if you want to only look at nuclear population, you have to bleach, let's say, the cytoplasmic one to see really the nuclear population dynamics, which can potentially be toxic for the mouse embryo. And as a matter of fact, it is. So what we then did is we looked at photoactivatable GFP. So this is a fluorescent protein introduced by Jennifer Lippincott-Schwartz and George Patterson, which also later was used by Eric Betzig for his Nobel Prize-winning experiments for super resolution. So this is a fluorescent protein which originally is not visible when you're excited with 488 nanometer light, blue light. But if you're excited with 405 nanometer light, so near UV, or violet light, then you can indeed then activate this protein, and with excitation of 488 again, you can render, for example, the nuclear population visible. And now we did not do 4 or 5, we did then pulse lasers. We figured out that pulse lasers of 820, so femtosecond pulse lasers, can do the same electronic transition, so the excitation. And so we activated OCT4 when attached to photoactivatable GFP in one nuclei within a cell of a developing mouse embryo, which was actually the first time actually applied under these conditions. And then we quantified the nuclear average intensity, and we could see them directly based on a so-called FDAP curve, so fluorescence decay after photoactivation or photoactivated OCT4. We could see after activation how the average intensity then dropped. And this could give us directly information about the export rate and the nuclear retention. What we accomplished there is prior to compaction, so we talk about 4-cell stage and 8-cell stage, we subjected blastomeres of single cells of this developing embryo to the FDAP measurements. And we could see that they fall into 2 clusters of distinct kinetic behaviors. As you can see on the right side, examples, and on the left side, you have the combination of all. So this was, regardless of other observations beforehand, we didn't look at shapes, mRNA, or cell divisions. A very new approach, dynamic imaging. And we really could see, let's say, cryptic differences, so not static imaging, but dynamic imaging, so these differences. I will cut it because it's not the purpose of this talk today, but what we figured out is that these early kinetic profiles, with some lineage tracing, we could see that they indeed also show us, let's say, propensity to allocate more to the inner cell mass or to the two-factor derm. So I'm going to go now, let's say, how this one evolved, let's say, from the technical side. On the molecular side, Nicolas Plachter was the first author on the story, has continued understanding more like the cytoskeleton mechanics, cytoskeleton molecular components, which are important also for lineage allocation. So he has really expanded this one tremendously. Now, what I found all the time a little bit limiting in this approach is that we're using photoactivatable GFP. And the problem of photoactivatable GFP is, as you can see, let's say in one of these embryos, is that you only have one single cell labeled and the other ones are not being labeled. And if you, let's say, want to have a transgenic line, let's say with CRISPR-Cas, all your transgenics are not visible. So based on screening, it's difficult to see them. And then even later on activating each without any cellular context, it's very, very difficult to appreciate, let's say, what's happening. Another limitation is if you look at single cells or subcellular, is that we just only activate a selective protein population. If you do a selective protein population, you already limit yourself in the interpretation of data. You only look at transcription factor binding to DNA, but you can directly see, let's say, nuclear cytoplasmic shuttling, let's say mechanics which influence the pore complex, the pore opening cannot be appreciated. The other one is if there are turnovers which differ between cells, you cannot really see them because you only indirectly can infer them based on these assays. So it would be, of course, nice to see all the different dynamics, turnovers, nuclear cytoplasmic shuttling as well in detail. So one approach to do, and this leads us to photoconvertible fluorescent proteins, is to use photoconvertible proteins. So what are photoconvertible fluorescent proteins? So these are proteins which are originally green, but with 405 nanometer light, you also can, in this case, not activate them, but shift their emission to red. So suppose here you have one of them photoconverted, so it's red, and the other ones then stay green, which allows you to analyze the cell, but still have cellular context. You can see everything. Obviously, a mouse embryo is a simple structure in comparison, let's say, to a huge tissue later where you have thousands and thousands of cells, where cellular context is even much more important. So, but it's nice to have all cells being labeled to track and infer also later on the development. If you look now subcellularly, you have the global view of all the protein populations, but you also can single out sparse protein populations based on photoconversion. So this allows you to, in one go, extract all the parameters. So you have a multi-parameter approach, turnovers, nuclear cytoplasmic shuttling, residence time. All these things can be analyzed. Now, we have done beforehand photoconversion. We did this the first time with Zebrafish. So this is a zebrafish which expresses a nuclear label. So it's histone attached to dendra2. Dendra2 is the photoconverted protein, a green to red one. And here you see the zebrafish embryo, all the nuclei being labeled. And a local illumination with 405 nanometer light can lead then also to a sparse labeling. In this case, when combined, you can see that beautifully you can do this nice illustration where you can illuminate just only the individual somites of a developing zebrafish embryo. Now, we also analyzed at that point in more detail the conditions to effectively or efficiently perform photoconversion. And here you see the laser power used for 4 or 5 nanometer light. And here we use the iterations in zebrafish. And you see the green dendra and you see here the red dendra. And what we figured out is that medium powers of laser, well, medium power settings of the laser and medium scan iterations actually led to efficient photoconversion. This is different to photoactivatable GFP. Photoactivatable GFP requires very short, high energy. So usually 100% laser power, if you just rough way, and just a few iterations are sufficient. The difference between photoconversion and photoactivation is illustrated here on the next slide. So in photoactivation, you also use 405 nanometer laser, but in this case, you have at position 222, you have a decarboxylation, and this leads only to an isomerization of the chromophore. Whereas in photoconversion, you really have a chain break. So you have a backbone cleavage. And this vibrationally potentially requires more extended exposure, but not to very high energies. Now, when we revisit the case of photoconverting now mouse embryos, for example, obviously I mentioned to you first 405. 405 is not the way of doing it, because 405, if you put 405 and you want to photoconvert one nuclear, for example, here with H2P dendra at the focal plane, the energy does not really drop above and below the focal plane. So you will also photoconvert then the nuclear, here in this case, below the focal plane. So one photon photoconversion is not actually confined. And here it's also being illustrated with dendra2 protein. And this one is immobilized in a gel. It's a cross-sectional view. We scanned here along the line. And you can see there's this hourglass shaped, reminiscent of the scan procedure of the laser. And you can see there is no confinement above and below the focal plane for dendritic photoconversion. Now, another way, obviously, to do confinement of photoconversion is the same as we applied for photoactivation. So using pulse lasers, near-infrared light, and focusing on one focal plane. This essentially has been shown previously that pulse lasers can induce an electronic transition, which is comparable to single photon, but just only at the focal plane. But you will see here that it doesn't happen. So you have dendritic protein, what we immobilized in the gel. So we tried here to photoconvert with pulse lasers with different wavelengths, as you can see here. And then we looked afterwards for the increase of red signal of dendritic protein. Here you can see the photoconversion fold. And as you can see, there is only an increase, very mild, in the range of 700 to 850, where we talk about 1.1 or 1.2 fold. And usually you would expect an increase of 20, 30 fold, for example. I hear other voices inside. I hope they can put the computer on mute.
You just have to hold it.
So
yeah, someone has unmuted them.
Here,
can we just mute the people, actually, please?
I know this from lectures.
Sometimes someone.
Okay.
I think it's okay now.
Yeah. So thank you. So as you can see, efficient two-photon photoconversion is not achievable, and you see the increase of the red channel for different types of classes of photoconverted proteins. They all stay rather flat. So this is for EOS family, Kaede family, Kiga family. You will see one increase here, which is 10-fold, but this one is a photoactivatable lamp cherry. So this is photoisomerization. So isomerization works with pulse lasers in a very short period of time, but photoconversion, which has the bond cleavage, doesn't work really. So I will now show you something, and I just cut now due to time now how we came to this one. Usually one says in science, everything is based on serendipity. This was not really that much. But it's something that's supplementary material, which actually the reviewers wanted to cut out, but we insisted on having it inside. So, but briefly now, what is prime conversion? So we did once immobilize dendritic protein. We illuminated 488 nanometer, the small area. It has been previously shown that Lyukhanov, his group, showed that 488 can mildly photoconvert. You can see a little bit of red. When you use only 730 nanometer, not pulsed, continuous wave, nothing happens. If you combine these two wavelengths, so blue and green, red, then you can actually have a pronounced photoconversion. We call this primed conversion. So the idea is you have a fluorescent protein in the green state, 488 is being absorbed. We identified this, that it comes to a transient state. I will explain in detail later on, but this transient state is not short. It's a millisecond transient state. So it's for milliseconds responsive to red light and to bring then the protein to the red state. Fluorescence usually is nanoseconds response time, and transient state here, it's more milliseconds. So we identify this one is potentially a different state. Now, the beauty of having two wavelengths is that you can also somehow combine them to accomplish primed conversion in a very precise volume. So in this case, confined. Imagine here the objective. Okay, this one is upside down. I should have done it differently. Sorry for this confusion. So this one is the lens. This is the back aperture of the objective. So light comes out here. The laser would come from top going down. So if you know that you have to use a blue light and red light, what you can do is you can put a filter set at the back aperture of the objective. And this will lead to a physical separation of both wavelengths, and they will only cross with each other at the focal plane. I have though also here a small separator in between, and let me explain you why we have a small separator, which actually accounts for this long prime state of a few milliseconds. So this movie is an oversimplification, but I hope it explains a little bit the idea. So laser comes from the bottom. You have this conversion filter plate, red permissive, blue permissive. You have this opaque strip, which doesn't allow anything. And then light should come through. And here is then the sample spot, let's say the focal plane. The movie shows you now that when light kicks in, It's being separated, and you can see that on top here you have them overlaying, but this separator here leads to, in this case, an insufficient temporal overlap here. So it's more than a few milliseconds delayed above and below, so that prime conversion only can occur at the focal plane. Is this true? Yes, it is true. And here you see the example. So you have a gel which immobilized Dendra2, a cross-sectional view, 4-5 laser, the traditional photoconversion laser. You see this hourglass shape when we just only photoconvert this line here. If you use prime conversion without any filter, also here you can see this hourglass shape. So you scan here, and then below and above there is also photoconversion occurring. If you use prime conversion in combination with a filter placed at the back aperture, you really can confine prime conversion to less than 5 micrometer now in this setup. Such a filter is available at AHF analyzing technique, and we have a Nature protocols which actually details how to implement it in a confocal microscope, for example. So what I would like to emphasize also is what kind of powers do you really use to confine photoconversion? Before with mouse, I showed you 2-photon photoactivation, and here you see that I put average power as well as peak illumination intensities, and you wonder why I put both. Well, Eric Betzig actually mentioned that what is the most important parameter for a tissue to experience and also to survive is not the average power, but the peak illumination intensity. So what it really is, the instantaneous intensity, what it experiences once the laser actually hits onto the tissue. And when you look on this one, you will recognize that 2-photon photoactivation actually comes here with 340,000 times more peak intensity values when compared to confined prime conversion. So with that in mind, prime conversion is not only confined, but due to the fact that you use continuous wave, it is also of lower energy, and this way also well below the tissue damage threshold of, let's say, lasers. In this way, we can apply to live animals. So the first what we did application is Zebrafish. So this live Zebrafish, it expresses Dendra2, the photoconvertible fluorescent protein. It's a pan-neuronal promoter, so QC. This one is the trigeminal ganglion, and this has this beautiful neuroids extending beneath the dermis of the zebrafish. And there are a lot of these ones clustered together. So it is very difficult to distinguish which neuroid actually belongs to which of these neurosoma. So what we did then is we focused on one of them, and we performed prime conversion as one soma, so the cytoplasmic Dendra2. This got then primed converted, but not the cells above and also not below. And given that it's live, it also starts to diffuse into the proximal neuroids. Now, this takes quite a while now because it is a diffusion-driven process to have sufficient contrast to build up, let's say, surface segmentation. But then you can really appreciate the beauty of this trigeminal ganglion and its proximal neuroids, which actually extend beneath the dermis of the zebrafish. And here you see then this illustration of real data where you can see then all these intricate structures of the neuroid then extending. What we also showed then is to perform prime conversion, obviously in mouse embryos. Mouse embryos are very, very sensitive. So 2-photon is not really something you should try to do for extended period of time, and especially not if you want to extract quantitative data out of it. So here you see on the lower one, again, H2P dendros, a nuclear label. And here we perform prime conversion. On the upper one, we perform standard photoconversion using 4-5. When you use prime conversion, so the combination of 488 and 730, you can accomplish photoconversion very precise in the nucleus without going through the entire nucleus, this cross-section view. However, when you use traditional 4-5 photoconversion, it is not confined, so it just goes right through the entire nucleus. One thing I should mention is, you see there's some bleaching going on here, which is not really nice, and I will come to this in a bit. Now, I showed you so far data for Dendra2. This is one class of photoconvertible proteins. We obviously try to do test also photoconversion other fluorescent photoconvertible protein classes, and one of them is the EOS family. But you probably can appreciate that once the protein is immobilized in a gel and will perform the prime conversion, the increase of its fluorescence, of the red fluorescence, is underwhelming, to say the least. So, and so we wondered, what's the difference between our ES2 and Dendra? So, and the other question we wanted to know is 730, when we present the first time, we got very much criticized, saying, okay, you generate something which is beautiful, but nobody can use it, because who has a 730 nanometer laser line? It's not a useful one. So we'll try now to address both questions: why the proteins don't work or not as good as some classes as let's say Dendra, and the other one is, can we combine it with laser lines which are more available in commercial microscopes? Well, the first one is to figure out what conversion beam, not the priming beam, which is 488, but what conversion beams really work. So what we did here actually is we immobilized again protein in a gel. We did the 488, the priming beam, and then we used different conversion beams. And then we used for this one white light laser. So we had different wavelengths, as you can see, from 600 to 825. Originally, we observed it with 730. And what we found out is that the peak actually is at 690. But interestingly, areas like 633 or 640 something, they are comparable to 730, which tells you that you can use a commercial system to perform primed conversion by combining 488 and 633, which are usually available in commercial systems. Now, we also wanted to understand, in this case, this prime state. I told you before, when you put 488, it brings it to a prime state, which is a long-lived state, 4 to 5 milliseconds actually, and then it goes over then to the red state. So what we did then is analysis in different media conditions. So what you see here is a semi-logarithmic plot. These are the delay times between the priming beam and the converting beam. In this case, we use 640 nanometer. So, and as you can see, the delay times up to the point where it worked actually gets shortened or extended. And this is based on different media where this protein was immobilized. So interestingly, the black line, what you see here, is actually air or water. So the normal conditions we use. If you then abolish oxygen, so you put this in nitrogen, then actually then the lifetime actually gets extended. But if you use, let's say, in this case, oxygen, or you use, importantly, in this potassium iodide, which is a triplet quencher, then actually then the lifetime of the prime state gets actually shortened. So with this one, we identified, because it's oxygen dependent, that potentially it goes through the triplet state through intersystem crossing. One point I should also mention is that we use deuterated buffer, and deuterated buffer here did not affect it in whatsoever. So it is like normal air or water solutions. And this tells you that excited state proton transfer does not occur actually. So the model of prime conversion is then as follows. It works on the anionic population of the photoconvertible protein. When you excite it, either you can have an emission in green, or you can have them from the excited triplets, single state, and intersystem crossing to the triplet state. This triplet state is a long-lived state, so in this case 5 milliseconds, and with red light, so as we know you can have a broad range, you can then bring it over to the red state, and it brings all the time the same final red species as if you were do it with a 4-5 photoconversion. Now, with the knowledge that it goes to the triplet state, we knew that there are photoconvertible fluorescent proteins which can, with their mutations, can influence, in principle, then the population of the dark states. And so we did a targeted mutation screen, and we identified one point mutation, and this was actually in position 69. And position 69, if it's threonine or if it's serine, here it's threonine, then it renders every photoconvertible fluorescent protein prime convertible. So Dendra 2 originally, so we were lucky, had on 69 the position at this position threonine and was prime convertible. However, EOS, the EOS family, the Kiga family, the Kaede family, and even Camparis or calcium integrators and calcium sensors like GR Gecko, well, Campari, they did not have really threonine serine, so that valine or alanine. And so by changing them to threonine, rendered them all of them prime convertible. And we call them in principle then PR, MESFP, for example. Importantly, with this modification, something else comes as an add-on benefit, and it's that they are more stable. So what you see here are then when the peptides have been illuminated with different powers of Void-8 laser, and this is the time half of the fluorescence get lost. And this is logarithmically now displayed here. And as you can see, the dashed line is the original MEOS2 protein. And if you now introduce this point mutation, you get here in this case a 6-fold increase in the green stability over the original MEOSFP. This is very critical because it tells you that photoconversion is limited. Why? Because you have a lot of bleaching before you can bring green over to the red one. Now, the red version is usually very stable already originally. And so this is also improved, but I think it's experimental, you will not really experience any difference. Now, with that in mind, we thought, okay, if we have such optimized fluorescent proteins, we should do super resolution. And we used commercial systems. So this was the Zeiss Palm system. So we used commercially first the conventional Palm, so 4 or 5 nanometer with EOS2. And we did also in parallel primed Palm, so Void-8 combined with 642. In this case, we tagged then a cytoskeleton protein with prime convertible MEOS2. So the advantage of this one is you can do dual color nanoscopy by having the same emission, but having different modalities of excitation. And here you can see the result. So this is the wide field. We have in one case, we tagged live actin with primed MEOS2, and then we have ensconsin tagged with the original MEOS2. And when we do primed Palm, we only then excite and visualize the super resolution structures of actin, and ensconsin structures are visualized with the traditional Palm. We're not the only ones who have done this. So prime conversion has proven beneficial in super resolution imaging. Ulrike Endersfelder, she has done multiplexing, for example, here, for example, fixed E. coli, where she labeled then RNA polymerase with a primed MEOS32. She did also live, this was fixed, and live here in HeLa cells, where she combined keratin dendra 2 with actin photoactivated M-cherry, where she also proved that this one has a much lower phototoxicity because it has a higher localization efficiency. And on the left side, just to give credit, this was Mishin's lab. They were the first actually to use prime conversion for super resolution. So this one is dendra 2 live again, live HeLa cells. Now this one is super resolution. And the other one, what we care about is also to look for benefits in volumetric imaging. So let's go back to mouse embryos. It doesn't have to be only for mouse embryos. Obviously, other tissues, zebrafish and organoids also could potentially benefit. So what we did here is to inject then RNA into mouse embryo with H2B primed MEOSFP. This is the protein we identified as the most optimal for mouse embryo of potentially for tissue imaging. Doesn't cluster in even high concentrations, is not toxic at all. So we then prime convert in a confocal microscope, again, separation and cross beam setup, and then we transferred them to a light sheet microscope to image it. Here you see now a movie at the fossil stage. So these are individual nuclei. So mind you, we prime convert them on a confocal, then we have to go then to a light sheet microscope. So there is some time delay. And as you can see, we were lucky to get them, let's say, before they had divided. So you have some condensation of chromatin already kicking in. So these are optimized proteins with H2B. So meaning they don't bleach much in green. All the population can be transferred to the red. We have high contrast. And the embryos are healthy and they develop nicely over the next 3 days of volumetric imaging. What you can see, cell divisions, you see more nuclei coming up. And later you can potentially even envision that there is the blastocells, so the opening up. However, there are two challenges. And one challenge is that you have spatial drift as well as rotational drift. It is important to keep this in mind because there are conclusions and track. If you really want to understand asymmetry, heterogeneity in tissue, you need to track each and every embryo. You should not exclude embryos. And I can tell you that the spatial rotational drift in a free floating system, in this case, so no agarose or something, is in the range of 45 to 50%. And previous publications have, in general, when they turned too fast or moved out of the imaging field, removed them from the analysis. And I do not think that this is valid if you want to do statistical conclusions in a complex system. So how can we then get these spatial rotations drift corrected? Well, we have prime conversion. It's not toxic. It's very precise label. So we use then the red color as a fiducial marker to orient the embryos at one direction and one orientation. So we define reference frames, let's say. This way we orient all the embryos at one side, the red one. We rotate them properly, we sample them, and we even crop them. This way we handle much less data. Mind you, dark data, still pixels, is still data. And this, of course, is a burden for handling volumetric data sets. When you put these together, obviously, you're going to have then, in this case, a cropped image, as you can see on the right side. On the left side, what you see is the pre-corrected movie. Now, these are the same movies. There's no change whatsoever. As you can see on the left side, there is no compensation for rotational and spatial drift. And on the right side, you see there has been, and you can believe me because around you see sometimes at the edges, some other embryos just disappearing and appearing again. Obviously, this one is, rather than using, let's say, massive of students and tracking each and every frame, this is, of course, a much faster, much more precise and smarter approach to do it. And this is actually what we thrive to do in our lab. So once you have this, you can do an automatization of fidelity. So here you see on the left side, before compensation, spatial rotational drift. We fed it with different algorithms, and we looked at the distance from the true corrected tree, the ground truth. And as you can see, from 4 nuclei on top, the system never, ever can accomplish the corrected tree. So it's not able to distinguish it if you just only feed the images as is. If you do a compensation, you can see something builds up a tree. However, you will also identify, well, Lucky's lab actually had to correct still to get it to the corrected tree. So the red signal 11 times and the green even 30 times. Why is that? Well, we did not perform, let's say, machine learning or some intelligence to the algorithm. The only analysis we did is when the cells divide, we have then spot sizes. And these spot sizes then identify nuclei, and the distance of these spots to the nuclei then gave the system a guess that these ones are daughter cells, for example. The system did not know that cell divisions in the mouse embryo do occur asynchronously. But however, they should still follow a particular path in some ways. They first have to divide before the other ones divide. So we had indeed then to identify the false connections, which was easily to be done because green and red, in some ways, we could identify when there was a false link. And then we corrected these ones. And by correcting these ones, we really then accomplished a total tree. Mind you, this is a 3-day movie. Computation, it takes a minute. You just press enter. The manual correction is still an hour. So from 3 days of imaging, it's a 1-hour analysis. Obviously, being not in pure artificial intelligence and also having the play, we hope then also to automatize this one, not only for mouse embryos, but also for organoids, where you have a much bigger spinning and even more nuclei. Well, these were movies done at 5 and 7 minutes frames, and you're pretty well aware that if you do light sheet, that you get huge data sets. And so it's even difficult to get them from the motherboard where you just do your imaging to some other computer analyze. So what we then thought is, if we're able to have such a high fidelity, why not imaging only every 40 minutes and see if you still can build up the tree? And as you can see here, indeed, from before compensation, it can build up some tree in this case, but with these corrections, we indeed can build up a corrected tree. And this tells us that we have less data to deal with. And this allows us also then to be fast in our analysis. Now, obviously here, this is rendering pre-prime track, and then you have the post-prime track. So we would love to see, of course, with this ability to track now these cells and their abilities, let's say their fate potential, either to go to the inner cellulose or to ectoderm, even let's say for cancer detection as well, if you want to do, let's say, metastatic or non-metastatic cancer evolution. So cell state in general would be nice to see whether they are, let's say, continuously evolving, or whether they have, let's say, also equipotential states during development. And now we have the way to have a comprehensive way to see all the nuclei at once, and in a precise way to tag whatever we want in some ways. Well, it sounds very optimistic in some ways because there are some limitations, and these are the final slides before I finish. So for prime track, there are also limitations, and these are the segmentation errors due to signal dilution. I mentioned to you we did prime conversion at one state, we have the red, then we move over to the light sheet, and then cell divisions kicks in. When you do prime conversion and you get your red signal, you decouple it from production. So the signal you have will get diluted with every cell division, given it's attached to the histone. They don't really get degraded. Histones have a very long turnover. In cell culture, over 200 days actually. But the more they get divided, the less the signal is intense. And sometimes you can have also false segmentations. For example, here, this is not a nuclei. This is autofluorescence, which has been identified by the system as a positive nuclear signal at a very late stage of the blastocyst. So we know that after 3 or 4 cell divisions, the signal competes already with autofluorescence due to dilution. So the challenge is, can we get something like an indelible label that does not dilute over time? Or the other one, a challenge would be, can we repeat prime conversion to prevent dilution of label? And let me show you both approaches, what we're trying now to establish. So in one case, I just showed you so far only passive labeling with prime conversion. There has been, and we worked together with Robert Campbell on optogenetics, so where we can even influence the function of cells by light, so optogenetically. And Robert came up with the photocleavable protein, so FOCO. This is a photoconvertible fluorescent protein. And upon prime conversion, this then also gets prime converted to red. But as you know, it has the bond cleavage. It has a second one. In this way, it then destabilizes, and then you have a spontaneous dissociation later on, loosen the fluorescence, and then you have, let's say, part of the protein then released. This, of course, the protein could be attached, for example, to a transcriptional factor, right? Transcriptional factor to activate some production gene expression. And indeed, this is what we did then. Here you see a cell with, I focus on in the lower one, primed conversion performed in the lower cell, cytoplasmic focal. It gets red. Later on, the fluorescence disappears. The small piece with transcriptional activator, then it's translocated into the nuclei. And then later you have a red nuclear expressed. This actually occurs when you have primed conversion. The same happens with 4-5, but does not happen, let's say, if you put the cells only in the dark. So focal is a nice tool, let's say, to control gene expression. And using, let's say, prime conversion. So what we did then is, or what we're actually doing now, is to use photobow. I showed you the beginning photo, which is a photoconvertible construct for zebrafish. And we want to combine it not only for prime converting a single cell, but also later on to have clonal lineage tracing, so clonal labeling, which takes advantage of the rainbow system. So the idea is you have photoconvertible nuclear label, it's H2B photoconvertible. And then once you put prime conversion onto your cell, you not only prime convert the nuclear, but you can also release Cre from focal. Focal is also co-expressed. So focal dissociates, Cre can then relocate into the nucleus, and then it can recombine in the different cassettes. And then you have, as a final outcome, you have then the cytoplasmic final color detections of red or green, let's say, or orange. Let me explain you this idea. So photoball would be initially a green nucleus, a green tissue with green nuclei. If you perform primed conversion, you then render, for example, 2 nuclei red, very precise confined prime conversion. In the meantime, you also split then focal, increase and release, let's say from the cytoplasm or the membrane, and translocate into the nucleus. This leads then to random recombination of these color color cassettes, and then you have, let's say, here green, and this one is more like magenta. Oh, this is more color, yeah, tissue color. So over time, then you could see if some cells are then dramatically then expanding. So obviously, then the nuclear label will be diluted and not visible, but it's still indelible label because of the recombination, or whether it did not dramatically then expand, and still you can see nuclear label. This is not science fiction. This is the photoball fish. You have here the nuclear, green nuclei, the cassette expressing prior to recombination. It's zebrafish 2.5 days post-fertilization. If you do systemic recombination, you can see that recombination occurs in all cells, the entire tissue of the zebrafish. We are also able to distinguish them. And obviously, we hope now to do systemic work in stereotypical development, but also in cancer evolution, for example. This is the idea, let's say, to address long-term lineage tracing. Another approach what we're trying to do is, as I mentioned to you, is to perform repeated prime conversion. So to combine primed conversion with a light sheet microscope. And here we teamed up with Chris Rowlands in our department in bioengineering at Imperial College London. So this one is actually a modified oblique plane microscope, which was introduced by Chris Dansby, also from Imperial. Here we have the 2 lasers, so 488 and 685. And these ones together then generate a crossbeam setup. So we can hopefully in the future track now measures of heterogeneities in embryos, organoids, tissues, not just in one, but in parallel settings, and also then based on repeating now prime conversion then with unlimited numbers of cell divisions. So as a summary, prime conversion is great for chemical interrogation of biological systems. So this means structure analysis, lineage tracing, and hopefully also with photoball. You can also use super resolution analysis and also optogenetic control. We hope to have them in the future. And one note now for people who are not aware of this one, 2 weeks ago, there was a big announcement from Leica and from Imperial that we have now established a Leica and Imperial College imaging hub, where we're gonna then establish, let's say, an efficient workflow of optical imaging, so capturing and also analysis. As I've shown you, for example, the ideas what I showed you for lineage tracing, where you use probes, what kind of microscope and what kind of analysis you have to do. And this we hope then to do. Workshops will be also being announced at some point, and also new systems will be then tested, and you can use them also and challenge them in some ways. And with this one, I would like to thank you for your attention. But before I finish, I would like to emphasize the guys who did the work. The person initially was William Dempsey, who was a postdoc, then who did the prime conversion. Manuel Moore actually figured out the protein engineering, so the different states, understanding the prime state, and also super resolution. Micah has done the lineage tracing in mouse embryos. Konstantinos now is evolving on primed photoball, as well as also on primed light sheet, prime conversion light sheet microscope. And critical collaborations on this work were Uli Nienhaus for understanding the prime state, Robert Kemper for optogenetics, and Chris is now for putting into reality a prime conversion light sheet. Okay, thank you, and I'm happy to take any questions.
Oh, thank you very much. It was a super interesting presentation, particularly for me, I must say. I discovered that we work on overlapping things, and I am very guilty that I didn't know that. So it was very nice to see your presentation. So we have a few questions for now, and I invite everyone to post questions to the chat, or even just to kind of book your slot to speak, just to kickstart the Q&A period. I just would like to mention perhaps one question from Stephanie that I see that you are already unmuted, so perhaps you can, although it was actually answered already.
Well, the answer was, yeah,
Yes.
when I was looking at the talk initially, I was asking about, can this be implemented with light sheet? And then the second half of the talk was all about that. So,
yeah.
So sorry.
So it's, well, I mean, we're still building the microscope on this one. The idea obviously is to do really precision imaging in a volumetric setup, biologist only. It's more to the question, actually. In some ways, we would like to have a smart microscope. So consider, for example, the biologist cares about this particular, let's say, protein dynamics within a cell, 1 point. Or the biologist cares about this cell where it moves in some way. So obviously, prime conversion can be very precise in getting rid of all the noise of other cells you would like to have. The tracking should be also democratized, so it's much simpler to do. But the other one, what we would like to do is also to have a smart microscope in its true essence, meaning when cells actually move during tissue, they don't move all the time in the same process and velocity. Sometimes cells stop, sometimes they get faster. So prime conversion, we do believe, can open up the ability to have an adaptable microscope, meaning that it recognizes when the cells move and then don't move, and then you can also have adaptive scanning in some way. So you really, this way, save scanning, save database, and you only extract information which is really, really critical for yourself. And this is actually what we would like to accomplish.
Because at the moment you need 2 microscopes, so you transfer from one to this light sheet and after the conversion. No?
Yeah.
So ideally
this
that
is the
would
first
be...
one. Once you have them implemented, then you can do it. And the second one is to have really smart microscope which can adapt actually to the dynamics you can see based on what kind of tissue you actually use.
So if you have like an open top light sheet, you could actually implement that, because that will be then open to additional photo manipulation. That would be great. Actually.
This is also, this is what we also try. So the idea is, that's the reason we do also what the original one introduced by Chris Dunsby. So first an inverted one, open one. We can do other modifications. It's very nice for mouse embryos, organoids, what other tissues as well. You can do parallelization of imaging with prime conversion. In the photo bow, we hope us to do then clonal expansions, whatever people would like to have. In some ways, also, let's say, tumor evolution is one option.
Yeah, no, very nice. We had Chris Dunsby as a speaker as well, so we have seen kind of a lot of his development. I think there's some questions directly in the chat now referring, so maybe
I postponed my own just to give the others. But if Nick, you were asking something about
the probes.
Yeah, I've got a couple actually, but one thing is just sort of how you can sort of multiplex with this technology. Do you think? I mean, are there sort of primable other photoconvertible proteins in the pipeline? Do you think? I mean, particularly if you think about live imaging, where people are starting to look towards the sort of far red fluorescent proteins as a good solution in that area.
So there are probably 2 things what we're trying to establish, but we're focusing on one, also limited with manpower in some ways. In one case, I showed you first that we first expanded 2 years ago a prime conversion to all photoconvertible fluorescent proteins by introducing this 1 point mutation. However, even under these conditions, prime conversion is much better. The proteins are much more stable, but we also know that going to the intersystem crossing is a very rare event. Yeah. So we need to improve this as well. So we are now doing also targeted mutations to improve it. Why do we do this? Because we want to do also prime conversion in a very reliable way with LEDs, because we would like to do, let's say, brain mapping. And so far, the power required on this one does work with LEDs, but it's at the edge in some ways. We try first to improve, also for optogenetics to go much deeper, for example. So this is a work with Konstantinos Kalibas, one PhD student, is actually exploring. We have preliminary, very good indication that we can induce it tremendously. So the second one is making prime conversion also for other fluorescent proteins. We do believe with the insight we gain now, we can potentially there are some indications that this can be a general principle of getting also, let's say, non-photoconvertible proteins even prime convertible. But we have not explored it really in much crazy detail. But this is something we are have started to work on. But I do believe that we have to understand first the prime state with this modification improvements more before we can go then to the other proteins with this as a guidance. But this will potentially also happen. Right,
great. Thanks very much. I just thought of one other question out of curiosity. So I think if I didn't miss this, most of your work has been priming with a sort of 730 or so nanometer laser. But you know, if I have a confocal in my hand, it's got a 633 or a 640 on it. I mean, how good are those lasers for this sort of application compared in your hands, would you say?
Yeah, so I showed it in the middle of the slides that we, of course, then analyze the different converting laser beams. And 730's quality is the same as 633 or 640. So they are comparable in the ability. So you can use them. All the super resolution images I showed you were done with a combination of 488 and 630 or 640.
Right.
And on commercial systems.
Right.
So, however, we know that, let's say, the best photoconversion is at 680, 690, but 630, 640 makes beautiful prime conversion. This is not a problem. So you can perform as we've done with 730. Now, why we have done 730 is because the coherent pulse lasers in the alignment modes are by default set up at 730. And we thought we required a lot of power. We don't require a lot of power. So what you require is a normal standard diode of 630. If you have it in your laser already inside, you can perform prime conversion easily. There's no problem.
Okay, great. Thank you very much.
Thank you very much. Next question is from Kevin.
Yeah, hey, excellent talk. So I think you talked about this to some extent, but do you have some idea of what the identity of that millisecond long intermediate state is? Is this some bond in the fluorophore that's maybe under strain and more susceptible to be broken?
I think this is one, I covered probably our failure really well in some ways, but so what I showed you are indirect proofs. So what we showed is, let's say, we knew number one is that the prime state is really in the range of 5 milliseconds. 5 milliseconds point. So it's a long-lived state. Now, obviously, to analyze this one, you could do it, let's say, in very low temperature. So you need to freeze the prime state. So, or you need to be super fast in some ways to analyze this. Under these conditions, we were not able to see because this was probably a low population. However, we haven't done NMR. So this is the one analysis we haven't done. However, indirectly, we have proven that with different oxygen conditions, where you put, let's say, your protein inside, the oxygen can really influence the lifetime. So it goes from in oxygen to 3 milliseconds, or it goes to 6 or 7 milliseconds in nitrogen. And if you put, let's say, potassium iodide, which is a quencher of triplet quencher, then also it gets lost dramatically. So we know it is going through an intersystem crossing, so to a triplet state. The second thing, what we also know is that there are no excited proton transfer, because deuterated buffers don't have any effect on the lifetime. So now there are 2 or 3 processes how the changes could happen and so on. I do believe that either by our new mutations we bring, which introduce a higher population of primes, so what we hope to have now. So we can potentially come to a place that we can analyze, let's say, with NMR or even with cryo, the population, see this directly what happens.
Okay, that'll be really cool.
Yeah.
Thank you. The next question is, I'm going to read it on behalf of James Walsh. He was asking if the point mutations that you're doing can cause any changes of the spectra for the converted fluorophores. And there's a second question, was asking if your plasmids are available on AdGene or something similar.
Number one, all the plasmids are available on AdGene. All the protocols also, everything is available in there. Also the softwares, I don't know if I put them there. I am happy to share the slides. The one thing is, now, prime conversion has the same end result as traditional photoconversion. So it has the same red fluorescent protein. So we don't have any change whatsoever in there. Now, the point mutation we introduce lead to changes in the stability. Definitely they lead to a stability change, also a little bit in the pH sensitivity. but significantly in the stability, and actually to a better one. So so we have up to 6-fold increase in the stability of the green version of this one. So in that sense, it's more beneficial. What we also tested in the different versions is which ones are good, let's say, for super resolution, and which ones are good, let's say, for volumetric imaging. I think I mentioned in the eLife paper we published 1.5 years ago, we used this primed MEOS-FP. So I suggest to use this one for volumetric imaging. EOS, Ulrike Endeslegger, she used primed MEOS 3.2. We used MEOS 3, I guess. Yeah, both are fine for super resolution, but they did not prove that fine for volumetric imaging, because in very, very, very high concentration, you have a little bit of dimerization.
Okay, great, thank you. So there is a question from Stanley Botchway. I don't know if okay. So I can read it for you. He was mentioning.
Hi, yes.
Ah, perfect, Stanley.
Yes, I'm here. Hi, no, thanks for a great talk, much enjoyed. So my question really is, I know you've used both 405 and 488 for the conversion, so plus the 630 to 780, wherever it is. How deep can you go into tissue in that case? Because you still have the low wavelength present.
Okay. It is at least, let's say, for organoids and for mouse embryos, it's not an issue because the penetration is sufficient. But you're right, let's say if you want to go much, 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 much deeper, obviously you have two things to consider. The limitation is not the converting beam, because this one is anyway already in the tissue transparency window. So we are dealing of 630, 680 or something. It is more the 488. So the question you ask can be also addressed, let's say, for other people who use 488. Now, in our, the difference is that we require for prime conversion very minimal 488 power and a little bit more power, 10 times more power for the red laser. So if we increase the 488, we can go dramatically more into depth. The other point what we're trying to do is, as I mentioned, is to improve the prime convertibility by other point mutations. And we have some indication that they are going by several fold higher. So we are very confident that we're going to probably have this millimeter prime convertibility even with LEDs.
Thank you.
Okay, thank you. We finished for now the questions posted on the chat. Of course, I have one or two questions. In the meanwhile, people could still post their questions if they want to. I'm particularly interested in optogenetics, and I wonder. I mean, already the use of focal is very interesting, and something that really is the main things I was referring that I neglected and I shouldn't have. But I wonder if you have any, how to say, ideas about the possibility of generalizing the mechanism of prime conversion also to other optogenetic tools that are not necessarily, you know, similar to fluorescent proteins.
Good question. So, I mean, probably some things. I mean, we are in constant contact with Robert Kemper because there is also, let's say, now an improved focal. So it's a focal 2, and we are also testing this now on prime convertibility, because the ability was before not outstanding. It was possible, but not outstandingly, especially for tissue applications. Yeah. Now, generalization. Obviously, what I mentioned before, can you use prime conversion for different types of non-modifiable, let's say, fluorescent proteins, rather than, let's say, photoconvertible fluorescent proteins? And can you use it for other? I do hope so, but I'm not certain of this. And we have some indications, but we are not there yet. So, and it's going to take some time, and we're happy also to collaborate on this one.
Yeah, I mean, it would be great to speak to you. I mean, we are doing similar things, but for different applications, and we wouldn't plan to work on photoconversion, but it's more a technique that we define as light dose painting, but effectively to exploit different type of non-linearities, more about the chemistry rather than the photophysics. But we have no idea if actually we work. It's only on paper at the moment, so it would be great to pick your brain of in another time.
More than happy.
Great. So Stephanie, I don't know if you want to add something, or we.
No, I think today I mean, sometimes we actually do a quiz and most of the lectures, which this time I didn't ask you actually because we were busy, but we will review this. And I think there were a few people who are waiting. We sent out these foldscopes to people. So because that was the lockdown joke, I guess, of the lecture series, that everybody who can't get access to the microscope should have a foldscope to play with. So I spare you the quiz today, so everybody will be pleased, because often it kind of goes a little bit wrong. But thank you very much for the talk. It was really excellent. It was absolutely amazing. So I think lots of people, as you saw, picked up possible collaborations. I don't think it was just Alessandro, but I think there was also lots of kind of interest in what you develop. And I mean, that's how, I guess with the Leica Excellence Center, there may also be opportunities for you to commercialize some of the ideas, or maybe you do that anyway. I don't know. I mean, it doesn't need to be Leica, but I guess I noticed you had quite a few patents actually for ideas. It was super nice.
It's being implemented. Oh, I shouldn't say it probably. It's being implemented there, but as an inventor, as first as testing case.
No, it's very, but it was very cool, and well, we hope you get lots of credit for the invention, and I mean lots of kind of research as well, obviously. So I don't have any more questions, but I'm sure if people want to ask you, and you know, they know how to contact you, and like that's the point of also creating collaborations and kind of across universities. So we're very, very happy that I'm very pleased, and thanks to your student as well to suggest you, to push you into our direction.
Okay.
So
thank you for the opportunity.
Thank you very much,
everyone. Thank you, Periklis. It was very nice to meet you and to see you.
Yeah.
Bye.
Sorry we can't go for a drink now, but
Bye-bye.
or lunch. But thank you, and hopefully we meet soon again in person.
Thank
Thanks.
you.
The kids are waiting. Bye-bye.
Bye-bye.
Thanks.
Bye. All right. Yeah.